Okay, the first part of this video is going to be studying protein transport by endocytosis. And endocytosis is the transfer of material from outside of the cell into the cell using the vesicle bound system. So endocytosis involves the formation of cytoplasmic vesicles by this infolding of the plasma membrane called invagination, and this occurs where there is something outside the cell that the cell wants to take up, typically via a receptor interaction, and then those receptors accumulate at a specific point on the plasma membrane. The plasma membrane folds inwards and then pinches off to cause or to form a transport vesicle. There are three main types. So phagocytosis, which is typically what macrophages will do when they come across something like a bacterium. They will phagocytose that and engulf it. There's pinocytosis that I'm not really going to talk about today, but that is just taking up water-soluble materials, also sort of commonly mentioned in textbooks as cell drinking. It's basically cells taking up water-soluble material in a small vesicle. And then finally there's receptor-mediated endocytosis, where a specific receptor binds a specific protein and that is taken up inside the cell. Now phagocytosis also technically does involve receptors of some sort, but receptor-mediated endocytosis tends to involve the uptake of a specific protein. So the part of the biosynthetic pathways that we're looking at today is this bit in green. So we're looking at the cell's exterior and getting material from the exterior of the cell into an endosome, and from that point onwards it can go to a whole range of different places within the vesicle bound system, as shown here. And over here you can see here, this is a bit of plasma membrane that's folding inwards, forms a transport vesicle. The material goes to the early endosome. That can be passed on to late endosomes and lysosomes. We'll talk about those today. Or that material can be passed on to other parts of the vesicle bound system, such as the Golgi stack, and then may be modified and sent elsewhere. So first of these is phagocytosis, literally cell eating, and this is where something like a macrophage will detect something foreign. So this could be either recognition of bacterial oligosaccharides like LPS, lipopolysaccharide. It could be directly detecting the FC portion of antibodies bound to something immobile, so such as antibodies bound to a virus or bound to a virally infected cell. Or it could be apoptotic cells, and apoptotic cells, when they undergo apoptosis, they expose phosphatidylserine, which is a phospholipid, on the cell surface, and that is a signal to be phagocytosed. Here you've got a bacterium attached to a macrophage, and we get these pseudopods forming around the bacterium, which then entirely engulf the bacterium, and we get the bacterium engulfed in a membrane-bound vesicle, and then we get the actin cytoskeleton forming around this. So this becomes an integral part of the cell. It is a vesicle that then can be fully internalized and sent deeper into the cell, typically to the lysosomes for destruction. And this is just an electron microscope image of that process going on. A couple of bacteria with pseudopods forming around the outside, and this is a phagocytic white blood cell that's engulfing this. Now a macrophage is perfectly able to entirely engulf a apoptotic cell. So yes, bacteria are sort of fairly small objects compared to a macrophage, but they will come along and take entire infected or damaged cells and engulf them and destroy them. Now the uptake of individual proteins tends to be done at what we call Clathrin-coated pits, and these are the vesicles that are used or initiated in endocytosis. So this image here is, if you imagine you're at the nucleus looking at the plasma membrane, you would see infolding of the plasma membrane with this hexagon polygon shaped structure to it, and this is a protein called clathrin, which is causing the plasma membrane to fold inwards. And this has been caused to fold inwards by a receptor-ligand interaction, which I will go on to in a moment. So these Clathrin-coated pits are formed by receptors for a specific protein, binding that specific protein, and when that occurs, adapter proteins are brought in and then that encourages a protein called Clathrin to bind, and that causes the plasma membrane to fold inwards, and that is how the vesicles are formed. And this is shown here on a cross section of some vesicles forming. So again, these electron microscopy images, this is the plasma membrane, this is the material that is being taken up by the cell. On here are receptors and Clathrin, and this is very electron dense. This is an electron microscopy image, and what you can see is over time through this sequence you've got what we call the invagination of the plasma membrane. At this point, formation of a transport vesicle which is almost pinched off, and then at this point we've got a transport vesicle which can then be transported throughout the cell. So Clathrin-coated pits are crucial to this, and Clathrin is a protein, and it's a protein that is 3 polypeptide chains of heavy chain and 3 polypeptides of light chain coming together in this 3-legged structure as shown here. And Clathrin proteins stick together in a very particular way, and what we can see here is this is one Clathrin protein with another one here in yellow and another one in green. So you can see how they link together arm to arm, and this creates this basket-like structure here. And when these link together, they naturally fold inwards to create the vesicle from a previously flat bit of plasma membrane. So it's this protein that causes the plasma membrane to fold inwards. And the Clathrin only accumulates where we need a vesicle to form. So this is the schematic of a receptor-mediated endocytosis caused by a receptor binding to a ligand. So here's the receptor for a protein, let's say low-density lipoprotein, which is a commonly uptaken protein. So the LDL receptor is expressed on the plasma membrane. When it binds to cargo, low-density lipoprotein in this case, that will cause the recruitment of some adapter proteins and then subsequently the recruitment of Clathrin. So the adapter proteins are in pale green, Clathrin is in dark green. So it's only when cargo binds a receptor that you get the Clathrin recruited. As more receptors bind more cargo, that brings in more Clathrin, and the Clathrin proteins start linking together, forming this basket-like structure here, which will eventually pinch off and create a transport vesicle which is coated in Clathrin and adapter proteins. Pretty soon after that forms, the Clathrin and the adapter proteins recycle back to the plasma membrane, and we have a naked transport vesicle which can then be moved to a different part of the cell within the vesicle bound system. So this is just shown in more detail and also showing the destination of these vesicles. So here's your LDL, the low density lipoprotein, that binds the low density lipoprotein receptor. That brings in the adapter proteins and the Clathrin, which forms a Clathrin-coated pit. That Clathrin-coated pit forms as a transport vesicle, initially coated, then it becomes an uncoated transport vesicle, and all of that Clathrin recycles back to the plasma membrane. This transport vesicle tends to then go and fuse with an endosome, and when it fuses with an endosome, the endosomes are a lower pH than the rest of the cell. That will cause a dissociation of the cargo from the receptor, so the LDL gets left in the endosome and the receptor can be recycled back to the plasma membrane to be reused. Then the low density lipoprotein can then be sent to the lysosome for degradation. That can be broken down into free cholesterol, it can be broken down into free amino acids and utilised by the cell in that way. And this whole process takes 10 minutes for a round trip for a receptor to go from bound to cargo to get to this area here and get back to the plasma membrane, and the Clathrin will recycle much quicker than that. Once the LDL ends up in the lysosome, once anything ends up in the lysosome, it will be broken down into its constituent parts, because the lysosome is full of hydrolytic enzymes that will break down almost anything. And this slide shows the fate of the various endocytosed molecules. It all depends on what the receptor is and what the cargo is. After uncoating, The vesicle will be delivered to an early endosome, which will be sitting just beneath the plasma membrane. This endosome is slightly acidic because it has ATP-driven hydrogen ion pumps on there, so it will have a lower pH than the rest of the cell. The cell's around 7.4. This would be less than 7. The early endosome can either fuse with the Golgi, if that's what's required, or it could be migrated to elsewhere in the cell, typically transported down the microtubules to fuse with a late endosome. A late endosome is a more acidic version of the early endosome, and then these can fuse with lysosomes themselves to form endolysosomes, and they then further mature back into lysosomes, where we end up with a very low pH and the ability to break down proteins, amino acids, carbohydrates, and break everything down to its basic building blocks, and that then can be reused by the cell if required. So endocytosed proteins can either be returned to the same area of the plasma membrane from where they came from. We saw that for the LDL receptor. The LDL receptor is expressed on the plasma membrane. It binds its cargo, it gets taken up into an endosome and then gets recycled back to the plasma membrane. So that's one fate of an endocytosed protein. The second one could be travels to the lysosomes for destruction, and this shows the difference in pH. The cytosol pH 7.2, pH of a lysosome is about pH 5, and it contains all of these enzymes, so anything that goes into there will be stripped down to amino acids, nucleotides, lipids and cholesterol. Phosphates will be stripped off, and everything can be recycled. And finally, material can undergo transcytosis, so this would be transporting material across a cell from one side to the other. And the sort of place you would find this quite commonly is blood vessels. So proteins that want to be secreted into the blood have got to pass through the endothelial cell layer that line blood vessels. So they would have receptors on one side, on the basal side of the cell, the receptor that would bind the cargo, that would be internalized, passed across the cell, and then be released on the opposite side of the cell, the luminal side of the cell, and release that protein into the blood. So transcytosis is the third fate of materials that are taken up, or the receptors. So this is the end of the first part. This is just a summary slide and a good revision checklist of all of the key points and definitions that I've talked about here, and you'll need this for the understanding of subsequent lectures on the vesicle bound system. Okay, so coming up next will be lecture 2 on this topic.